So good afternoon. First of all, I'd like to thank the organizers for the possibility to come here and present the data we've been doing in Groningen for the past, let's say, 4 years. I understand the competition was actually very tough, so thank you for that. And talking about competition, my talk today is about the mechanisms that are underlying the relationship between biodiversity, the biodiversity invasion relationship for soil microbial communities. And by the end of the talk, I hope I will convince you that competition, especially for resources, is the major mechanism driving this interaction. Right, so I would like to start. How does it work? Yeah. I would like to start by putting microbial invasion in the context of an invasion framework. So you can see it's a very simple framework. It consists of 4 different steps. Introduction, when an alien species arrives into an environment, in this case it's soil, but could be anything else. Then the second step is the establishment of these invading species, the spread, and the impact. This invasion framework can be seen from the invader's perspective, and there are some characteristics that are from this invader that make it more prone to invade new areas, and these characteristics are the high growth rates or higher dispersal abilities. But you can also imagine if you have invaders with a higher phenotypic plasticity and higher genetic diversity, these invaders will have a higher probability of adapting to a new environment. This framework can also be seen from a community or environmental perspective, and the idea behind is that communities, native communities with a lower diversity or environments where we have a higher resource availability, they are much more prone to be invaded. One important thing about this framework is that the native communities play a very important role, especially in the beginning of this framework, and they act as a biotic resistance, either blocking the establishment of the invader or the spread of these species. So putting all these things together, David Tillman, about 10 years ago, talked about the stochastic niche theory, but he summed up all this stuff saying that the greater diversity should lead to lower invasion probabilities, and the idea behind is that community invisibility will depend pretty much on the degree in which the established species utilize the resources that are available, habitat heterogeneity in the limiting resources. So basically what we wanted to answer was whether soil microbial diversity would actually act as a barrier against bacterial invasion, and why? That was the major question. And the hypothesis that, based on Tillman's work, is that if you have a higher diversity, then you should have a higher competition rates, and that would lead to lower invasibility. So you can also look at like this graph, where you have in the x-axis the resource supply and the y-axis the resource uptake. And you can imagine whenever resource supply is higher than uptake, then you fall within this yellow triangle. That these communities where you have this condition, they should be more susceptible to invasion simply because they have a higher resource availability. So you can also think, put the diversity within this context, and you think of communities with a lower native diversity, they probably have very low resource uptake, and in that case the probability of having a higher resource supply is higher. In the case of communities with a higher native diversity, then probably they have a very high uptake, and in this case they have their competition is very high, and they are much less prone to be invaded. Right. We are also interested in understanding this part of the framework, so specifically how invasion affects the soil native communities, but for the sake of this talk, I'm going to focus on the first part of the framework. Right. So our invading species is a very common bacteria. It's called E. coli. We all have it, and I hope we have the good ones, not the pathogenic ones. And there were 2 main reasons why we decided to use E. coli for our studies. One of them, well, first, is that, as I said, is a very common organism. It's found in the gut of many vertebrates. And the second very interesting characteristic of this species that actually it is invading natural ecosystem very often. So actually every time an animal defecates, E. coli has the ability to get into a new ecosystem, new environment, and its ability to survive actually determines its ability to recolonize a new host, so to close its own cycle. So we were interested in understanding what would happen with E. coli, especially this very nasty one, the 0157H7, when it falls into the soil, and whether the diversity of the soil microorganisms could actually act as a biotic resistance. So a couple of years ago, we published a paper where we showed that diversity indeed act as a biotic barrier towards pathogen invasion, in that case, a bacterial pathogen. And this is one of the pictures from this experiment where we show that E. coli survival decreases a long time for certain treatments. And the treatments here are actually recreated. Those are gradients of diversity, and they were created pretty much the same way that Laurent described this earlier this afternoon, using the removal approach. So what you can see basically is that when you have in treatments with a lower microbial diversity, E. coli survives much longer than the treatments with a higher microbial diversity. What we did not answer from this work was what were the mechanisms driving this relationship. So that was the challenge that Cyrus Mellon, who is here drinking wine in Lyon, this was the challenge of his thesis. He started about 4 years ago, and we wanted to test the hypothesis that was, well, microbial diversity is negatively related to invader survival, but moreover, we wanted to understand why. And our major idea was that resource competition would be the major driver. So in order to do that, we created different communities. And we used two different approaches. One is the assemble approach, and the other is the removal one. That's the one that Laurent mentioned before. So for the assemble approach, we had a pool of 75 soil strains. The soil strains, they were collected from different places in the Netherlands. They had been characterized by 16S and by metabolic profiles, and also by box PCR. And what we did was actually pretty simple. We created communities varying in terms of diversity, so species richness. And we had communities containing 5, 15, 30, or 60 species. And you can see that we also have some replicates here, and these replicates are actually compositional replicates. So we have 24 different communities containing 5 species, for instance. And the idea there is that you can really separate richness from composition. Right, but you can say that 60 species is not really very representative of what we actually have as diversity in soils. So, and I will have to agree with you. So for that, to get closer to the real diversity, we used this removal approach. So we took a soil sample from Wageningen in the Netherlands, a grassland soil. We made an initial dilution, and from there we made serial dilution, so basically the removal or dilution to extinction approach. Once we'd done that, we took these dilutions as well as these communities, and we inoculated them in a sterile soil sterilized by gamma radiation, and this soil was coming from this region. We are also interested in looking at changes in composition, also using the removal approach. Therefore, we took soils also from a different area, in this case a potato field in the Netherlands, and we made the same approach. We diluted, and we reintroduced that in the same soil W, which we had here, to avoid any problems in terms of differences in soil type. Right. What was very nice about this approach is that we managed to create a very broad diversity gradient that vary from sterile soil, 5 species, up to 100, you see 500 species. Okay, so once these these communities were assembled, we had to let them establish first before we could actually inoculate our pathogen, and this took about 30 to 60 days, depending on the approach. But once we reached normal CFU levels, so about 10 to the 9 cells per gram of soil, we introduced our pathogen, the E. coli strain. And we then did a couple of measurements to follow the fate of this E. coli through time. And these measurements are shown here. And we looked at E. coli abundance and bacterial abundance by doing total counts on specific median or general media, but we also looked at the same data using qPCR. We were interested in understanding the metabolic potential of these communities because we think resource competition is very important, so therefore we used metabolic diversity, and for that we used a biolog system, the plate gene3 A, which has about 72 different carbon sources. And we looked at bacterial diversity by sequencing the 16S ribosomal RNA gene of all these communities. Right, so go to the first results. The first thing we wanted to see is whether the removal approach had actually worked. So here is an example of this W soil, and you see the dilutions are here, and you see that indeed by diluting we had a shift in composition. That's something we were aiming for. And also we saw a reduction in diversity, especially from the -6 compared to the other treatments. But what you see, and that's something that's very interesting, is that the -1 and the -3 treatment, I actually have very similar levels of bacterial richness. You can also see that for the B soil, we have a similar pattern, but we have a different composition compared to the W soil. Right, then what about the survival patterns? So these are the results from the assemblage experiment and the removal from the W and the B communities. And if you focus first on the blue line here, you see that in all the experiments, whenever you add E. coli to a sterile soil, E. coli tends to survive for pretty much the whole duration of the experiment. However, when we introduce this E. coli in a soil that contains, doesn't matter how many bacterial species, can be 5, but can also be 500, you see that it tends to decline with time. We then wanted to know whether that was really dependent on species richness, and this graph is summarizing the data from the assemblage and the W soil, and you see that there is a very nice negative correlation between the abundance of E. coli at 28 days and the species richness in the soil. So indeed, richness is is acting as a biotic resistance against E. coli abundance or E. coli survival. Right, but this is just confirming the results we had before, and what we wanted to know is why we have this pattern. So as I mentioned before, we were looking at the metabolic profiles of the bacterial communities, and we also had the metabolic profiles of E. coli. So what we did next was to try to determine the potential of the communities, the resource uptake potential of these communities. So we simply overlaid the data from E. coli with the data from the bacterial communities, and you see that there are some sources that are used only by separate communities, some are used only by E. coli, but you also have those that are used by both. So what we did was to remove these from the analysis, and what we get is something like this, where you have here a number of a pattern, and I forgot to mention that these colors here, if it's white, it means that the source, that the community is not capable of using these specific sources, and if there is a color, it means that the community is capable of doing so. In our case, we were a bit more, we also looked at the intensity, but it's not represented here. Right, so what we did was to take these values here and calculate what we call the community niche, and this is a metric I developed together with Franck Poli and Xavier Leroux in 2009, and this community gives an idea, this index gives an idea of how the community can actually exploit the resources that are available in the environment. So the idea is, if we have a community with a higher community niche, then this community should be able to compete for resources in a more efficient manner than a community with a lower community niche. And therefore, if we correlate that with survival, we would expect a negative correlation. And that's exactly what we showed, is that for the assemblage experiment and for the removal experiment, we have a very nice negative correlation. We also looked at the communities the sources that E. coli could use, and we see that indeed when we have more sources that E. coli can use, we have a positive relationship, so E. coli survives better, which was kind of a nice but obvious result. Right, so then we show that when we have a higher resource uptake, we actually have a graph like this, so the communities with a lower diversity we see E. coli surviving, but once diversity is higher, this survival also decreases. But all I showed before was just about correlation, so we wanted really to prove what was going on. So we decided to do something like this. We decided to shift the community from this point here to a point where we have enough resource supply. And we did that by adding resource to the communities. And the idea was that if resource supply is really driving this, the invasibility of the community, then we should have the two communities being invaded by E. coli. So this is the results. We did that only for the assemblage experiment. And so this is what I showed you before. And about 95 days after E. coli was inoculated, we then add D-galactose, and this was a resource that's being used both by the community and by E. coli. And if you look, for instance, at this red line here, you see that the full line is the treatment 5 species where we added galactose, and the dashed line is the treatment where we add water instead of galactose. And you see that E. coli really increases in abundance almost to levels close to the beginning of the experiment. So we then correlate that with the ability of the community to compete for this galactose, and we found, as expected, a very nice negative relationship. And what was also very interesting is that we saw that, so this is a relationship between the species richness in the community and the survival of E. coli during this period after we added galactose. And we see that there was still a very strong negative relationship between species richness and survival, despite the addition of the galactose. Right, so I see that the light is red. So I'll finish up. So the take-home message, why is soil microbial diversity acting as a barrier against invasion? So I hope I convinced you that competition for resources is the reason why these communities are acting as biotic resistant or being acting as a resistance. We also showed that, and an important point is that we showed that for a very broad gradient, diversity grading, going from very simple communities but also to more complex ones. We also saw that resource addition releases the competition in these communities, and that this release of competition is still controlled by diversity to a certain extent. So regarding the next part, I cannot, of course, present you the results, but I can tell you that we have very interesting results. I hope I can talk about them in another time. Okay, so with that, I would like to thank the microbial ecology group in Groningen, especially Cyrus, who did 99% of this work, but also Frank, Poli, and Xavier, who received Cyrus for about a year, some time ago, and I would like to thank you for your attention.
Thank you. Questions for Joana?
Thank you. That was a very interesting talk. I was just wondering, and I don't know how strongly you feel about this, but in any experiment in which you are increasing diversity and you are even replicating with different communities or compositions just to try to tear apart the effect of composition, you're still increasing the probability of, in the higher diversity treatments, to catch the few, maybe, it's a possibility, the few species that are actually highly competitive. So imagine a community where you have quite a few, but not very many, species that are very capable of competing out E. coli. Mm-hmm. If you increase diversity in such a long gradient as you have, you are also increasing the probability of catching one or two of them. So how do you tear apart from this?
Yeah, so this is indeed a possibility. What we see is that when we get from, well, these communities, they are reintroduced in soils, right? So they actually, those that can respond much rapidly are those that we are seeing at the end, the moment where we inoculate E. coli. So those are indeed the most competitive ones. Yeah, it's hard to tease them apart. It's already hard enough to manipulate microbial diversity and get something, that makes sense out of it. We tried to look at the importance maybe of other mechanisms, not only competition, and see whether that would actually affect the results. And so we looked, for instance, for antagonism between the communities, and we could not find any correlation. So it might be that we are selecting for competitive species. That's true, but it's very hard to get rid of them.
Another question at the back, please.
I see that the survival of the E. coli decreases only at high diversity level. That is, the treatments that are basically in the soil dilution area. What is the contribution of potential biocontrol agents compared to the resource competition hypothesis in the survival of E. coli that you introduced?
So maybe for the first part of the question, actually E. coli decreases in all the treatments, also in the 5 species treatment. It's just at a lower rate, but it decreases through time. And about the second part, I'm not sure I understood the question. So you're asking what would be the contribution of this theory for the biocontrol agents?
Exactly, because resource competition is one aspect, but the biocontrol could be completely different. It may not be nutrient competition for nutrient, it may be bacteria being engulfed by another bacteria and or other organisms.
Yeah, so I think in terms of biocontrol agent, this theory is pretty much applied to biocontrol, to addition of biocontrol species to the environment, and I think it might explain also why most of the biocontrol agents don't really tend to survive in soil once they are inoculated. It's simply because they are arriving into a very high community with a high diversity, and there's a lot of competition. So I saw from your side that I didn't get the question. Maybe.
I'm not talking about introducing biocontrol, but when you made the dilutions, there may be existing biocontrol agents in the high diversity community.
Yes.
Just it's kind of building on the question that was asked over there.
Yeah, so we did not find any biocontrol. So from the assemblage experiment, we really had communities where they had a higher number of species that have the possibility to control E. coli in vitro, and we see no effect of that on the survival of E. coli. On the removal experiment, we didn't look at that. What I can tell you is that after, so we looked at how the community changes with time after the invasion, and what we see is that in the communities where E. coli decreases, we see an increase in Pseudomonas species, which could indicate in species that are capable of producing antibiotics, but this is completely, it's based really purely on correlation.